The next section we're going to focus on is the results section. And so in this section, this is where you'll find a lot of the data that was collected for the study. Descriptions of the data will be found in the main text of this section, as well as immediate interpretations of the data. Now, this section, of course, is where all of the facts of the study are presented. And because of that, what I typically suggest is that you don't read the results section, at least not until you've looked at the data, identified trends in the data, and try to interpret the data yourself. And this is important. This skill is important for critical analysis of any scientific paper. And of course, if you're just starting out, this will be difficult to do because you're not familiar with the types of experiments that are used, or how data are presented, or the background of a particular study, so that you can actually interpret the data in the context of the broader relevance. But with practice and time, you will get better at this. And like I said, this is a very important skill to have for reading papers. So what this video is going to do is describe how you might write a description of a figure within a research article. So you could do this with either your own data, or you can do this with data in a paper that you're reading. And in the latter case, what's nice about that is you can write your own description of the data and then compare it to the authors and see if you both came to the same conclusions. So here's a figure from a paper. The citation is down here for those of you playing at home. So here's the data set that's shown. So this is called the time course. So as you see, the x-axis is in time, and then the y-axis, there are actually two y-axes. So there's one on the left-hand side, which is concentration, and so this refers to the concentration of this molecule called NG, and also nitrate. On the right-hand y-axis, this is OD600, so this is known as optical density. So this measures accumulation of cells within a culture. Okay, and so when you're looking at data in a results section, what you should be able to do ideally is you can see the figure and use information from the caption. And if the caption, in an ideal situation, the caption will be written such that all the information necessary to interpret the data is written within the caption. Okay, so the caption down here says growth of Variovorax species strain JS1663 on NNG as the sole carbon, nitrogen, and energy sources. So this is just a description of what this piece of data is showing without providing any interpretation. Squares, optical density of 600 nanometers, right axis, so this is what we described earlier as describing what the axes are. Circles, nitrate concentration, and diamonds, NNG concentration. Okay, so we have some information as to how to look at these data. All right, so there are a few things we're going to want to look at. And again, hopefully, ideally, most of the information you would need would be within the caption. So we're going to start with what is the experiment. So we know the experiment. That's from the first line. It's Variovorax species strain cultures using NNG as the sole carbon nitrogen sources. And we're monitoring both nitrate concentration and the cell density. So the caption includes all this information. In some cases, this may also be, you may also need to visit the materials and methods, which typically has a more detailed description of the experiment in order to figure out what you need for this. What is being measured? And more importantly, how, right? So again, the caption sufficiently describes that cell density is being measured, NNG concentration is being measured, and nitrate concentration is being measured. So in this case, in the caption, it doesn't say anything about how any of this is being measured, but this is adequately described in materials and methods. And so NNG is being measured by LCMS or HPLC. Nitrate concentration is being measured by a grease assay, and then OD600 as the name suggests, is being measured by UV vis absorption spectroscopy. So are controls shown? This is really important because without controls, we don't know if the positive results that we see are real or are due to some other effect other than, in this case, the bacteria consuming the NNG. So there are no controls shown in this figure, and you may have to look elsewhere in the results section, where the results section may say something like controls of NNG decomposition in the absence of bacteria, for instance, which would be an appropriate control for this study, showed no decomposition of NNG or formation of nitrite. And so that may be something you may look for in the results section. This just may be written without actually showing a figure. Data. So now we're looking for trends in the results. And so what we want to see is just adequately describe what the trends are. So we'll start with NNG concentration. So NNG concentration starts to decrease from time 0 to 15 hours, and then it's completely consumed. Then we can look at nitrate concentration. And so we follow the line for nitrate concentration, and this starts to increase all the way up to about 20 hours where it about flatlines. Then finally, for the OD600, there is no measurable OD600 until all the NNG is consumed, and then it starts to increase, going up to about 50 hours, where it plateaus. All right, so this is all you're trying to do here is just describe the trends that you see. Now interpret and compare with the authors. So now take all the trends that you see, all the information that you know, and write a short description of the trends that you see, and then you're going to compare it with the authors. If you have sufficient information about the background information for this, then you may also be able to try and interpret it in a broader context or make immediate interpretations of the data. So for instance, for the data shown, we have what I've written is, for instance, this may be something you might write for a description of figure 1 in the results section. So green, the green highlighted, indicates exactly what we discussed, just the trends seen in the data. So figure 1 shows time courses monitoring N-nitroglycine, NNG, and nitrate concentrations and OD600 cultures containing varivorax species strain JS1663. NNG was monitored by LCMS and nitrite was monitored by grease assay. So this indicates how these two molecules were monitored to get these data. Time 0 represents the time at which the culture is inoculated and NNG was added, whereupon the NNG concentration decreased with all the NNG consumed after 15 hours. There is a concomitant and stoichiometric increase of nitrite during this time. The OD600 starts to increase after the NNG is completely consumed. So for this is just a description of figure 1, highlighted in green. The red concerns the control, and so this is just something I'm adding. This may not be what is actually shown in the results section of the paper, but this is just something I've added about the controls. An appropriate control would be decomposition of energy in the absence of varivorax species strain JS1663, or in the presence of another bacteria such as E. coli. Finally, and highlighted in yellow, would be the interpretation of the data we've just described. Assuming NNG is not decomposed under these conditions, the time courses suggest that varivorax species strain JS1663 decomposes NNG to form nitrite as one of the products. Another product that the bacteria can use as a carbon source must be made during this decomposition to support the observed cell growth after 15 hours. Okay, so we have this, we can write this out again. We can just do this in our own words, describe the figure, and then compare it to what the author said. And that may, if we draw the same conclusions, then we can reasonably feel confident in the interpretation of that figure. And so in the paper that this figure is from, here's where the section where it discusses figure 1. And so they say, much more succinctly than I did, that strain JS1663 released stoichiometric amounts of nitrite during aerobic growth on NNG as the source of carbon. The disappearance of NNG and release of nitrite were complete before growth began. So that's very similar to what I've said in the paragraph that I wrote. Now in yellow, this is their interpretation of those of that description, which indicated that an unidentified denitrated intermediate accumulated and served as the growth substrate. Since nitrite accumulated during the disappearance of NNG, we hypothesized the initial cleavage reaction might be between the amino and nitro groups, yielding glycine as an intermediate. However, glycine supported only weak growth, data not shown. So this is interesting. So their interpretation of this data went much further than what than anything that was written in my paragraph, where they posited that that glycine might be a product upon decomposition of NNG. And so they did that, allowed them to create a new hypothesis in this study for a new experiment. And they just simply described that glycine really, their experiment using glycine had only weak growth, and so that didn't support this hypothesis that glycine was one of the products of NNG decomposition. So they just say data not shown, but they explain it. But again, like I said, there may be some parts such as control experiments or other sort of follow experiments such as this that result from the figures you're looking at that may not have an actual figure. And so it's helpful then to read the results section eventually, but I still suggest that you try to interpret all data on your own before reading the results section. Now,